Ribosomes are tiny machines that build proteins inside every living cell. They're made of two parts, a small subunit and a large subunit. The small subunit holds the mRNA, which carries the instructions from DNA. It binds to the mRNA and reads its code, 3 letters at a time. The large subunit works like a simple assembly line. It brings in transfer RNA, or tRNA, which carries amino acids. To understand how this assembly line works, we need to look at 3 important sites on the large subunit. These are A site, P site, and E site. The A site, or aminoacyl site, is the entry point. A new tRNA arrives here carrying a specific amino acid. For example, in this case we have methionine. Each tRNA has an anticodon, a set of nucleotides that must match the codon on the mRNA. If the match is correct, the tRNA is accepted into the ribosome. Like here, codon on mRNA is AUG, and anticodon on tRNA is UAC. Next is the P site, or the peptidyl site. This is where the growing protein chain is held and extended. A new tRNA enters the A site, another amino acid is added to the chain, and the tRNA that has already done its job moves to the E site, the exit site, and leaves the ribosome. This cycle of A site to P site to E site repeats again and again, adding 1 amino acid at a time, forming a growing amino acid chain. This chain later folds into a functional protein, and then the ribosome releases it, and the protein goes on to do its job in the cell. Also, not all ribosomes are the same. Ribosomes are mainly classified based on their sedimentation behavior, not their size or weight. This is why ribosomes are described as 70S and 80S. The letter S stands for Svedberg unit, which tells us how fast a ribosome sediments when spun in an ultracentrifuge. 70S ribosomes are found in prokaryotic cells, like bacteria. They are made of a 50S large subunit and a 30S small subunit. 80S ribosomes are found in eukaryotic cells, like plant and animal cells. They are made of a 60S large subunit and a 40S small subunit. One important thing to remember is that these values are not additive. So, 50S plus 30S does not become 80S, and 60S plus 40S does not become 100S. This is because the Svedberg unit depends on sedimentation rate, which is influenced by mass, shape, and density, and simple addition is not enough. How exactly this S value is calculated is a topic on its own, and I'll be explaining the calculation of Svedberg units in a separate video.